RNA transcription is the process by which a gene is expressed into an mRNA strand. For some genes encoded, RNA is used to synthesize a protein via gene expression. Expression can be divided into two processes, transcription and translation. DNA contains chromosomes, and chromosomes consist of millions of base pairs, some for which code for a gene. These genes code for specific proteins. DNA is wrapped around alkaline proteins known as histones. Active areas of DNA are wrapped less tightly than other inactive areas, so to be more accessible for RNA polymerase to bind and begin transcription. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends with a terminator. When a gene is expressed, a specific protein is produced. The process of transcription occurs in 3 stages. Initiation. The promoter region of a gene functions as a recognition site for RNA polymerase binding. Gene expression can be controlled in this way by either permitting or blocking access to this site by RNA polymerase. This binding causes the DNA double helix to unwind and open. One strand serves as an antisense strand and one strand serves as a sense strand. Elongation. RNA polymerase moves downstream along the template strand, synthesizing the mRNA as it goes. Reading the antisense strand in a 3 to 5' direction, it generates the mRNA from the 5' end, attaching RNA nucleotides to 3' end as it goes. This occurs via complementary base pairing. RNA is synthesized instead of DNA, therefore it's a ribose sugar, not a deoxyribose, and uracil is utilized instead of thymidine. Termination. Once RNA polymerase reaches the terminator portion of the gene, the mRNA transcript is complete, and they dissociate from each other. The pre-mRNA must first be processed by the addition of a methylated cap and polyadenylated tail. 3' end of the mRNA is moved into the correct configuration for cleavage. Stabilizing factors are then added to the complex. Poly-A polymerase now binds to the mRNA and cleaves the 3' end. Poly-A polymerase then synthesizes the polyadenylated tail by adding adenine residues to the cleavage site. When the tail reaches its full length, the poly-A is signaled to stop adding residues. The processed mRNA is now ready to undergo splicing. Intron splicing. The strand of messenger RNA that's made during the transcript includes exons that code and non-coding sections called introns. In order for the mRNA to be used in translation, the introns need to be removed. This process is performed by a complex of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce the mature mRNA strand. This mRNA strand is then able to leave the nucleus via a nuclear pore, ready for the next step, translation.